Photosystem II, PS2, and Photosystem I, PS1, are two major components of the photosynthetic electron transport chain, which also includes the cytochrome complex. The cytochrome complex, an enzyme composed of two protein complexes, transfers the electrons from the carrier molecule plastoquinone to the protein plastocyanin, thus enabling both the transfer of protons across the thylakoid membrane and the transfer of electrons from PS2 to PS1. The reaction center of PS2, called P680, delivers its high-energy electrons, one at a time, to the primary electron acceptor, and then to plastoquinone. P680's missing electrons are replaced by extracting a low-energy electron from each of two water molecules. Splitting two molecules is required to form one molecule of diatomic O2 gas. As electrons move through the proteins that reside between PS2 and PS1, they lose energy. This energy is used to move hydrogen atoms from the stromal side of the membrane to the thylakoid lumen. Those hydrogen atoms, plus the ones produced by splitting water, accumulate in the thylakoid lumen and will be used to synthesize ATP in a later step. Because the electrons have lost energy, they must be re-energized by PS1, hence, another photon is absorbed. That energy is relayed to the PS1 reaction center, called P700. P700 sends these high-energy electrons to ferredoxin, which sends them to NADP reductase. NADP reductase passes an electron to NADP+ to form NADPH. NADPH is used in the Calvin cycle. Thus, PS2 captures the energy to create proton gradients to make ATP, and PS1 captures the energy to reduce NADP+ into NADPH. The buildup of hydrogen ions inside the thylakoid lumen creates a concentration gradient. In the thylakoid, a specialized protein channel called the ATP synthase allows the protons to pass out of the thylakoid. The energy released by the hydrogen ion stream allows ATP synthase to form a molecule of ATP from ADP and inorganic phosphate. The flow of hydrogen ions through ATP synthase is called chemiosmosis because the ions move from an area of high to an area of low concentration through a semipermeable structure of the thylakoid.